Good morning to all. Myself, Dr. S.K. Aminabi, Coordinator, Head Department of Pharmacology in BB Institute of Pharmaceutical Sciences, Gudlavaleru, Andhra Pradesh. It's my privilege to invite you all to the two-day e-conference on alternatives to animal experimentation in pharmacology, organized by Department of Pharmacology, BB Institute of Pharmaceutical Sciences, Gudlavaleru, in association with ANU College of Pharmaceutical Sciences, Nagarjuna Nagar. First, I would like to thank Professor A. Premila Rani Madam, Principal, ANU College of Pharmaceutical Sciences, Nagarjuna Nagar, and Professor A. Lakshman Rao Sir, Principal, BB Institute of Pharmaceutical Sciences, Gudlavaleru, for their continuous support in organizing this e-conference. Let's begin this e-conference with a prayer song. Various alternatives to the use of animals has been suggested, such as in vitro models, cell structures, cell cultures, computer models, and new imaging and analyzing techniques. The in vitro models provide the opportunity to study the cellular response in a closed system where the experimental conditions are met. Such models provide preliminary information for the outcome of an experiment in vitro. For example, computer models were used to study the working of the heart as well as to study the function of the heart as well as to study the
Resounding.
Resound is coming.
Okay, sir. With this brief introduction to the
Madam.
Lakshman, it is resounding. Amina.
Yeah, I'll continue. Yes, madam.
Okay, madam.
Various alternatives to the use of animals have been suggested, such as in vitro models, cell cultures, computer models, and new imaging analytical techniques. The in vitro models provide the opportunity to study the cellular response in a closed system where the experimental conditions are maintained. Such models provide preliminary information for outcome of an experiment in vivo. For example, computer models were used to study the working of an heart and to select the potential drug candidates. With this aim, we have organized e-conference on alternatives to animal experimentation in pharmacology. With this brief introduction to e-conference, now I request Professor A. Lakshman Rao Sir, Principal and Convener, BB Institute of Pharmaceutical Sciences, Gudlavaleru, to talk few words about this e-conference. Over to Lakshman Sir.
Thank you, Amina Gandhi. Respected Chief Guest of today's e-conference, Senior Academician, my Guru and Mentor, Professor A. Premila Rani Garu, Convener of the e-conference and Principal, University College of Pharmaceutical Sciences, Acharya Nagarjuna University, Nagarjuna Nagar, and eminent speaker of the today's session, Dr. Madhav N. Mogale Garu, Senior Scientist, Department of Toxicology and Experimental Medicine, Central Drug Research Institute, CSIR Lab, Lucknow, and Dr. Sheikh Aminabi Garu, Coordinator for the e-conference and Head Department of Pharmacology, faculty and student participants from various colleges. A very good morning to one and all. It's my privilege to welcome you all for the two-day National e-conference on alternatives to animal experimentation in pharmacology, organized by Department of Pharmacology, VV Institute of Pharmaceutical Sciences, Gudlavalleri, in association with University College of Pharmaceutical Sciences, Acharya Nagarjuna University, Nagarjuna, on 3rd and 4th September 2020. And this program is will be more beneficial for teaching fraternity as well as student community in the area of pharmacology. First of all, let me thank Acharya Nagarjuna University authorities for giving acceptance for association with us to conduct National Online E-Conference. On behalf of management, principal, staff and students of VP Institute of Pharmaceutical Sciences, we thank Honorable Vice Chancellor, Professor P. Rajashekar Garu, and Rector, Professor P. Varaprasad Murthy Garu, and Registrar, Professor K. Rosey Garu, and Principal, Professor A. Premiladani Garu, ANU College of Pharmaceutical Sciences, Nagarjuna, for their kind support for organizing this e-conference. I am happy to inform you that our college, VV Institute of Pharmaceutical Sciences, Gudlavalleri, was sponsored by AINM and VV Adisar Educational Society, Gudlavalleri Post Krishna District, Andhra Pradesh. And we started this institution there 2010 and running with both BPharm as well as M-Pharmacy, Pharmaceutics and Pharmaceutical Analysis specializations. And ours is one of the prominent premier institute in the pharmacy education in the state of Andhra Pradesh. I proudly announce that our institution, VV Institute of Pharmaceutical Sciences, Gudlavalleri, is conducting this e-conference program for 2 days with 2 plenary lectures and with 2 senior research scientists as resource persons in collaboration with a renowned state government university, that is Acharya Nagarjuna University. And our previous activities were successful in conduction of the series of the events like webinars, international faculty development program, and online short-term training programs in various departments of our college in association with various universities as well as various social bodies also. Okay, and I hope this particular we came before you to for these 2 days online e-conference. On the occasion of this e-conference, we conducted e-poster presentations competitions also for the participants. We got very good response from the participants, and results will be announced in valedictory function, that is on tomorrow. And the e-conference, I am happy to inform you that the e-conference proceedings and full-length articles will also be published in the UGC Care List Group 2 journal, that is Journal of Interdisciplinary Cycle Research. And the major objectives of the today online e-conference program are to enrich the knowledge on various alternatives to animal experimentation in pharmacology. The number of animals used in research has increased with the advancement of research and development. Every year, millions of experimental animals are used over the world. The pain, distress, and death experienced by the animals during scientific experiments have been a debating issue for a long time. The organizations like ICH, CPCA, NIH, and OECD provide various guidelines for animal testing, and various in vitro models are available, and these are considered as an alternative to animal experimentation. And the examples include the computer models and cell cultures and tissue cultures and various alternative organisms. And nowadays, the computer-assisted learning techniques is a major alternative to the animal experiments, and these are having a greater advantage over animal experiments because of the regulatory and ethical issues. And the program schedule includes, in day 1, Dr. Madhav N. Mogale, Senior Scientist, Department of Toxicology and Experimental Medicine, CSIR, Central Drug Research Institute, Lucknow, will deliver a lecture on alternatives to animal experimentation in pharmacology. In day 2, that is tomorrow, Dr. Pradeep Chakraborty, Senior Scientist, Department of Assisted Reproduction, ICMR, Institute of Reproductive Medicine, Kolkata, will deliver a lecture on pubertal metabolic and endocrine changes. Part 2, Adolescent Polycystic Ovary Syndrome and Unexplained Pregnancy Loss. I strongly believe that this online e-conference will become an astonishing conference, and I wish all the participants will gain the knowledge on alternatives to animal experimentation in pharmacology. I am happy to inform you that totally 918 participants were registered for this 2-day e-conference program from various reputed pharmacy colleges in India. And thank you all for your active participation with your scientific temper during the COVID-19 pandemic situation. I request all the participants to attend all 2 days plenary lectures and gain the scientific knowledge in the area of pharmacology. Once again, I specially thank Professor A. Premiladani Garu, convener of the e-conference. Although her very busy schedule with a lot of domestic problems, for sparing her valuable time for our e-conference as a chief guest for the inaugural session of our e-conference program. Now, I invite the today's chief guest and also a convener of second convener of this particular national e-conference, Professor A. Premila Rani Garu, to address the participants. Thank you, one and all, and over to Dr. A. Premila Rani, madam. Madam?
So, am I audible? Am I audible, Lakshman?
Oh, it is audible, madam.
Okay.
Oh, okay, madam. It is audible.
Okay. Okay. Thank you. Thank you, Professor Lakshman Rao Garu, Professor and Principal, VV Institute of Pharmaceutical Sciences, Gudlavalleru, Dr. Amina B, Head of the Department of Pharmacology, faculty and student participants from various colleges, and also the resource persons of today and tomorrow sessions. A very good morning to one and all. It's my privilege to welcome you all for this 2 days e-conference on alternatives to animal experimentation in pharmacology, organized by Department of Pharmacology, VB Institute of Pharmaceutical Sciences, Gudlavalleru, in association with our college, that is ANU College of Pharmaceutical Sciences, Nagarjun Nagar, and today and tomorrow, that is 3rd and 4th September 2020. And first of all, let me thank Professor Lakshman Rao, who was my student, Professor and Principal, and Dr. S.K. Aminabi, Head Department of Pharmacology, VB Institute of Pharmaceutical Sciences, Gudlavalleru, for their ideas to organize e-conference in which there are guest lectures by the eminent speakers and e-posters, video presentation and publications of articles in UGC Care List Group 2 journal, that is Journal of Interdisciplinary Cycle Research. And computer assisted learning techniques as an alternative to animal experiments is having a greater advantage due to increasing difficulty to perform animal experiments because of issues, so many issues related to the procurement of animals, strict regulations, and ethical issues. And previously, the practical classes in pharmacology for undergraduate pharmacy courses in India included more than 15 animal experiments using frogs, mice, rats, guinea pigs, and also rabbits. At present, the animal experiments have been replaced by alternatives with computer-aided animal experimentation, computer-aided techniques. Typically, these techniques can fulfill the learning objectives of B.Pharmacy and PharmD students to a greater extent. And I strongly believe that this e-conference on alternatives to animal experimentation in pharmacology will become a grand success, and I wish all the participants will gain wide knowledge on this. And once again, I thank Professor Lakshman Rao, convener, and Aminabi, coordinator of this e-conference, and faculty and students' participation from various colleges. And I personally feel it's our opportunity and my honor to involve in this prestigious e-conference as co-convener. In fact, our registrar, Professor Rosai Garu, wanted to be associated with this today, but due to very urgent meeting with the higher education department, he couldn't able to participate, and he wanted the same to convey through me, and always the blessings and support of Acharya Nagarjuna University administration, Vice Chancellor, Rector, and Registrar blessings are and support is with VV Institute of Pharmaceutical Sciences, and particularly the principal of VV Institute of Pharmaceutical Sciences, Lakshman, is doing very great job during this pandemic COVID-19 outbreak. I came to know that there are several e-conferences and webinars and faculty training programs, student training programs. They have been conducting from March onwards, and really I'm very happy. It's great of you, and my special congratulations to principal and also team VV Institute of Pharmaceutical Sciences, and wishing many more in future also. And the main objective of any type of and any e-conference or seminar during these days is to engage students with a perfect or equipped with some knowledge on a particular topic. And because of this reason, VV Institute of Pharmaceutical Sciences conducting so many webinars, and this one is about pharmacology, particularly the alternative techniques, apart from alternative techniques to animal experiments. Really, it's a very good topic, and I request all the participants to be involved seriously to get a very good knowledge on this, which is a burning issue. And we have received so many letters from Pharmacy Council of India also to implement and to follow alternative technologies and techniques to animal experiments. And though we have animal house, we don't use animals for animal experiments for BPharm undergraduate students. And whereas for M-Pharm and research purpose, animal house it is being used. So with these few remarks, I once again express my deep felt of gratitude, and I feel really it's my honor to be associated with this prestigious university run by prestigious uh, the authority, which is having a very good reputation in the society, not only in Andhra Pradesh but also in India. So once again and finally, I wish all a very good success to this two-day e-conference, and congratulations to the team VV Institute of Pharmaceutical Sciences, and also to all the participants presenting to this seminar. Thank you very much for giving me this opportunity. Thank you, Professor Lakshman. Thank you very much.
Thank you very much, madam, for your gracious presence on this auspicious occasion. Now I invite Dr. Sheikh Aminabi, coordinator for this e-conference and head department of pharmacology for the e-conference, to introduce the today's speaker, Dr. Madhav N. Mugale Garu, to the participants. Over to Dr. Sheikh Aminabi.
Thank you, sir. Thank you, A. Lakshman Rao, sir, and thank you, A. Premila Rani Madam, for your kind support in extending to organize this e-conference. Now I take it's my privilege to introduce today's speaker, Dr. Madhav N. Mugale, Senior Scientist, Department of Toxicology and Experimental Medicine, CSIR, CDRI, Lucknow, Uttar Pradesh. Sir was awarded with PhD in Veterinary Pathology from NBC, Chennai in the year 2015. He published 27 articles, out of which 11 are international and 16 are national. He guided 3 PhD students and 3 PG students. He delivered 18 guest lectures and attended 24 training programs. Sir received Young Scientist Award in the year 2016 from Indian Association of Veterinary Pathology, Chhattisgarh. PhD Best Thesis Award in the year 2016 from Indian Association of Veterinary Pathology. Young Scientist Award in the year 2015 from Indian Association of Veterinary Pathology in Gannavaram, Andhra Pradesh. Molecular Oncology Award in the year 2014 from Anand Agriculture University, Gujarat. Sir is a reviewer for scientific journals like Journal of Veterinary Science, Comparative Clinical Pathology. He is a life member of many scientific societies like Veterinary Council of India, Indian Association of Veterinary Pathologists, Maharashtra State Veterinary Council, Society of Toxicology of India, Laboratory Animal Science Association, Laboratory Animal Science Association of India. With this brief introduction, now I hand over to Dr. Madhav N. Mugale, Senior Scientist, CSIR CDRI, Lucknow, Uttar Pradesh, to continue the session. Over to the speaker. Sir,
Mogale garu, start your session.
Mughal sir. Madhav sir, can you hear me, sir?
Amina garu, start is joining.
Sir, okay, sir.
What happened?
Sir, madam,
Hello?
just 2 minutes, madam.
Madam,
Madam,
problem in the SAR side, madam. He is joining, madam.
Okay, no problem, no problem.
Madhav sir. Dear participants, there is some network issue from the side of the speaker, so bear with us for a couple of minutes. There is some network issue with the speaker, Madhav sir, so he'll be joining in a couple of minutes. Please bear with us. Madhav sir? Madhav sir? Sir, can you hear me, sir?
It is audible, Amina garu.
Sir, that's sir. Network problem.
Hello, principal sir. There is a network problem from star side.
Madhav sir? Hello, sir.
PPT వచ్చింది డౌన్లోడ్ చేస్తా.
Yeah.
Sir, sir.
Hello. Hello?
Yeah, is it audible?
It is audible. It is
Yes,
audible.
sir. Yes, sir. You are
So
audible.
start.
And backup for backup we send through WhatsApp also. We don't know what will. ठीक है, ठीक है, खोल दो। चेयर लगा दो
Sir,
this one.
can you see the
Yeah,
screen, sir?
yeah.
Yeah. Okay, sir.
Now you got the screen.
Ah,
Yes, sir.
yes, sir.
Yes,
You
sir.
can
We can see your
enlarge the
screen,
screen,
sir.
sir. Shift F5. Enlarge the
Thank
screen.
you, thank you,
Okay, sir.
thank you.
तो ये लैपटॉप ये चार्जर लेके आप इमेज पे बैठ जाएंगे ना तो आपको परफेक्टली काम करेगा।
कहाँ?
ये इमेज जो है सर
हाँ हाँ
वहाँ बैठिए सर काम करेगा ठीक है।
ठीक है ठीक है. Hello. Good morning, all of you. Sorry for disturbance. It's my pleasure to be part of this e-webinar by the Institute of. Hello, is it
Sir,
audible?
it is audible, sir.
Yeah.
Yes, sir. It is audible, sir.
Please down the camera, sir. To your face, sir. Oh, okay. Oh, it is good. It is good.
Uh, it's my pleasure with the part of e-webinar and by the Institute of Institute Pharmaceutical Sciences, Gudla Veluru, Krishna district, Andhra Pradesh, and special thanks to management of the university and Dr. Lakshmana Rao Garu. Thank you, thank you. I will start my webinar Alternatives to Animal Experimentation in Pharmacology. Okay, we can start now.
Now you can start, sir.
Yeah, thank you. Uh, Currently, drug discovery is the very important in these emerging diseases. So there are steps are involved in the early discovery, pre-clinical studies, clinical development, and for the FDA review and for post-marketing monitoring. But during the drug discovery, we are doing hundreds of projects from the initial stage to from the chemistry to the marketing. But each and every steps we are using some animals and also humans in a clinical trials. But for this, there are lots of ethical issues are coming and species life we are uh, spending in this experimentation. So nowadays there is a in vitro models in drug discovery is a very important for the innovation of the lead molecules or drug. So first we have to do the target by in vitro models. We have to identify the target identification and validation of the study. Then you should lead identification and optimization. Then we have to do the ADME study, absorption, distribution, metabolism, excretion, and toxicology assessment. We will go one by one. So, what is a model for in vitro or alternative experimentation? A model, a representation of an object, a system, or an idea is in some form other than the entity itself. Due to cost, ethics, and precious human or animal life, it's difficult or impossible to do the study directly in the humans or animals at the preliminary screening level. So, alternative model is the best for the screening of the drugs at the preliminary levels. So, there are different types of models are there: physical model, mathematical model, and biological models. We will go one by one. Currently, in the study, there are millions of animals are used for the development of the lead molecule or candidate drug. In the current scenario, 74% of different strains of mice are used for the experimentation. There are bug, Swiss, Webster, C57, and knockout mice is used. And even some studies, rats, after that, then fish, and other mammals and amphibians are also used. So, To save the millions of lives, there is a need of alternative alternative models for experimentation. So the animals are having the life. They are also precious. They have right to the life. So we should use animals or we should not use the animals that we have to plan before the study, and we should avoid to use the animals directly. There are different rat, mice, guinea pigs, dogs, and even some large animals such as monkeys, dogs are used for the study of the lead molecule in the various stage levels. So, all over the world, there is a very big uh, animal lovers, pet are different yeah, different NGOs. NGOs are there to they are telling uh, to stop the animal experiments. Then there are also argument is going on the all over the world. If we stop animal research, we will stop the real killers. Currently, there is a pandemic of corona is also going on. So some are saying I would not be alive without animal taste. Animal research saved by my mom. There are lots of things are going all over the world. Also, they thanks to animal research, they will be able to protest 20.8 years longer. They saved my moms. Like this, there are those are telling. So, animal experimentation is also important, but the more than that, alternative model discovery is very important. So, for the experimentation, animals lowest on the phylogenetic scale should be used for this step and use minimum number of animals to get statistically valid results at 95% degree of the confidence. Alternatives should be used wherever possible, should not go directly in the animals. Disadvantage of animal experimentation: pain, distress, unethical behavior to animals, requirement of skilled manpower required for animal experimentation, animal experiments are time consuming protocol, high quality is involved in that, translational rates of the animal experiments are extreme.
Madhav sir,
Yes, sir. Yes, tell me. Yeah, tell me.
sir, participants are saying that your voice is not clear, sir.
Voice is not clear.
Yeah, yeah.
Wait, just. Now it's clear.
Yeah, yeah, now it's clear, sir.
So, advantage of the alternative models are direct use of human tissue in toxicity is more reliable than animal toxicity. So, during the experimentation, we can use different flies, fish for the experimentation. Abhinav, you can listen. Hello, is it audible?
Sir? Yeah, yeah, it is audible, sir. Sir, it is audible.
So, during the animal research, the alternative model discovery is a very important. So, there are the guiding principle of 3R: replacement, reduction, and refinement by given by Birch. So, we will go by replacement or alternative models. First is by using the cell culture. Aminavi, is it audible? Hello?
Yes, sir, it is
Hello?
audible, sir.
Yeah. So, by first is by cells or tissue culture. In this, we will use either primary cell line or commercial cell, and there some specific Special instruments are required, such as biosafety cabinet, basal tube facility, inverted microscope, CO2 incubator, and also liquid nitrogen for the storing of the cells. Pri- Primary cells is the cells which is derived from the direct organs, and commercial cell lines, which is the specific cell line derived by the manufacturer and they will supply. So by use of this, we can do the cell line study, such as in our lab, we are working on the liver cancer cell line, HEP-G2, A54 cell line, A549 cell line, on lung cancer epithelial cell lines, HeLa cell lines, and HeLa cell line is the cervical cancer cell lines of the humans, and human embryonic kidney cell lines 293 we can use for the screening of the various drugs as a experimental models. Also in primary cell culture, we can isolate the beta cells from the pancreas, and we will see the efficacy study of the different anti-diabetic molecules by analyzing the secretion from the beta cells such as insulin, glucagon. So we can these we can observe the cytotoxicity assay, ROS assay, reactive oxygen species, and mitochondrial membrane potential assay. These are the some of the cell lines from my labs. DLD1 colon cancer cell line, fibroblast cell line, and NCI lung cancer epithelial cell lines. We can do the experimentation in at the preliminary stage level on this cell line to save the precious life of the animals. Second model is the zebrafish model. Well, there are lots of institute having the zebrafish facility, because zebrafish having uh, they can uh, gives the number of the fishes fingerlings, also easy to handle, lifespan is small, so that we can do the number of studies. Zebrafish model generally used for the regeneration of heart tissue, tail regeneration, retinal degeneration, human pigmentation, cancer research, and autism. So, geographies are now being used virtually all disciplines, from the neurogenesis to oncogenesis, from behavior to genetic. Its a popularity is steadily increasing in current drug discovery. So, this in a lot of institutes such as TIFR Hyderabad is having, IISA Bangalore is having, and even our institute, uh, the fish facility. So second model is a honeybee. Honeybee generally used for the ecotoxicity studies. If honeybees and their flourishness is in our area is there, so you can say the pollution is less. Honeybee are commonly used as a sensitive indicator of air contamination. For 1 kg of honey, honeybee visits 15 millions of blossoms, and they will collect the nectar from this, and uh, due to this visiting of 15 millions of the blossom, they will bring at the same time some samples from the air, also from the flowers. And we can on that we can test the toxicity of that area. Also, from the honey, the propolis, a kind of glue used for the sealing and also for the inflammatory model development diseases. So, propolis with the tincture iodine used as a hearing impairment also. So, honeybee is also a good model for the development of the hearing impairment study, and also inflammatory study and ecotoxicity study. Fourth is C. elegans. C. elegans is the worm. The phylogenetic level is very less. It's invertebrate, and it's used nowadays lot of in the drug development. Of the C. elegans, there are three Nobel Prize winners. Winners are there in the history in 1998, 2002, and 2006. So, C. elegans model generally used for the neuronal development such as depression, psychosis, Parkinson's disease, Alzheimer's disease, pain model. Also, some nerve tissue injury study also. And even metabolic disease type 2 models, obesity studies, uh, also in aging, short lifespan helps to conduct a genetic screen to find the longevity of genes uh, because the C. elegans we can prepare in our culture plate, in our lab, we can prepare plenty of the C. elegans by providing the specific culture to grow and replicate. Also, it's used as a cardiovascular, oncology, and muscle disease models. In our institute, C. elegans facility is also there. One of my colleagues is working on the C. elegans. So, in C. elegans, the scientists had developed uh, and published the papers on programmed cell death 1982. Complete curriculum neuroscience was established in 1986. Complete genome they are studied. First use of GAP in animals. In Nobel Prize in 1994 they won. Also in 2008, and first animal whose whole genome was sequenced. Fifth model is earthworm. Yes, I had worked on an earthworm model for the ecotoxicity. So this is also like maybe like we can use as a sensitive indicator for the ecotoxicity, specifically for the soil contamination, soil pollution. Everybody we knows that earthworm is a friend of farmers. When there is a pollution of the soil, there will be the less number of the earthworm will be there. Or when we do the study, we can take the representative soil from that area and we can culture the earthworm and we can see the effect of this particular drug on the earthworm or also cocoons or pupae of the earthworms. I have published one paper on this earthworm. Six is invertebrates such as Drosophila, fruit fly. Also, we can use the for the mutagenic study, teratogenic study, and the reproductive toxicity study. So, also sea urchin used for the basic reproductive research. Butterfly. Butterfly are sensitive to the changes in habitat climate which influence the distribution and abundance. Useful tool in the study of the genetics. In CCMB Hyderabad and IISc Bangalore, some scientists are working on the butterfly, especially in the genetics. Genetics. Lal test, no, popularly known as Lal test, Limulus amoeboid lysis test, and monocyte activation test. Based on the human, also uh, from the human leukocytes, based on the response of the human leukocyte, which release the inflammatory medicines in response to the viral contamination. In your rabbit test, Limulus amoebocyte Lal test, the whole blood pyrogen test method was first discovered by Altex exactly 20 years ago. In vitro pyrogen test is internationally validated and it is a replacement to the uh, rabbit test. So we are by the Lal test. Now what is Lal test? In this, the liposaccharide causes extracellular coagulation of blood and there will be secretion of the blood and cytokine and excretion from the blood vessels, which leads to uh, the stiffening of the blood vessels. We can see by three techniques. By this latest gel clot technique based on gel formation, turbimetric method based on development of turbidity after the cleavage endogenous substrate, and chromogenic method based on development of color, the cleavage of synthetic peptide chromosome. So we can use it as a in vitro parasitic testing as a, will be as a lal test. Monocyte activation test. In this test, the blood from the human blood, monocytes are isolated, and that monocyte, monocyte is indicator for the chronic disease model. From that monocyte, when we will test the drug, there is an activation of the monocyte. Monocyte will secrete different kinds of interleukins such as interleukin 6, which leads to the increase in temperature or pyrogen, or increase gives the signal to the brain, and if that level, if you check, will indicate the yes, there is a there is a if this test item you use, there will be the increase in the temperature of the particular person. So detect the pro-inflammatory content is better than lal and rabbit pyrogen testing. The monocyte activation is very popular nowadays. So this is the things. Sample from the human or animals will be taken. Whole blood uh, will be will be processed through for a procedure, and primary monocyte will be collected, and different interleukin cytokines and TNF and neuropterin will be detected by the commercial tools. Then there is a embryonic stem cell test used for the embryonic toxicity studies. In this, the stem cells from the rodent cell line D3 will be collected, and it will be observed for the inhibition of differentiation, cytotoxicity by the MTT assay, cytotoxicity effect on the 350 fibroblasts. For toxicity also, we can use uh, this one, the stem cell, embryonic stem cell test. Local lymph node assay. In this, the lymph node from the humans or animals will be collected, and if test compound is tested against the lymph node, lymphocyte will be isolated from the lymph node. When lymph node draining the site, chemical application will be done, and if there is a proliferation of lymphocyte, there is a in- increase in number of lymphocytes in that uh, place, and there will be secretion of some chemokines. If there is a proliferation proportional to the dose, then you can say there is a stimulation of lymphocyte and increase in stimulation index. And if it's more index must be at least 3 for this also. Skin patch test. Nowadays, in the market, there are lots of creams are coming to test to protect against pollutions, to protect against different skin diseases. For this, first we have to do the skin test, phototoxicity study, uh, or skin patch test. Is it, when we will apply to the skin, is it leads to any corrosive action or itching, or it's safe to the skin? So for this also, there is no need to use the antigens. So in this, which what we will do, we will isolate the some skin epidermal keratinocytes, and that keratinocyte we, we will culture in the plate, and After some incubation period, the cells will begin. We should provide some nutrients such as media, such as mixture of water, sugar, amino acids. And after the 10 days, you will see there is a increase in the and proliferation of the keratinocytes. After 10 days, we can ultraviolet light. We can uh, give the light, focus on this UV light, and we can see the parameters such as swelling is there, redness is there, and all these things. This is a very important test. It's replaced the rabbit raised rabbit skin irritation test due to the skin patch test. Same, which I had just now mentioned the things. Neutral red uptake assay. For the eye ocular different ocular solutions, the generally this study is done by the rabbit eye. And due to this neutral uptake assay, it's replaced the rabbit eye test. We have reduced the use of rabbit in uh, uh, eye testing. Neut, uh, in this, neutral red penetrate cell membrane and accumulates in the intracellularly in lysosome. Alteration of surface of lysosomal membrane results in decreased uptake, eh, and if there is a decrease in uptake, the decrease in color of the things. This is the cells which is isolated from the eye of the rabbits and it's grown. These are the stellate cells. You can see the spindle cells. And if there is a decrease in the red uptake, you can say this drug is safe for the use as a ocular solution. Some of the studies are carcinogenicity testing. In cancer, we are using different cell lines. For the animal models, by using the cell transmission assay, BALC 33 assay, Syrian hamster embryo, we can use as a carcinogenetic testing. These assays are faster, less expensive, and involve fewer animals. So, using the cell lines and also the hamster embryo, we can use as a uh, carcinogenetic testing we can do. The hence egg test, polyoallantic membrane test. In this, the growth of blood vessels we can check. Even yeah, in this vaccine development also CAM test polyoallantic membrane used. What we do in this, the eggs will be incubated for 21 days. During the this 21 after 21 days, the chicks will be prepared by the eggs by providing the temperature and also ventilation. So when we will incubate the eggs in incubator at the day 12 to 14, there is a development of angiogenesis in the eggs, and we can this angiogenesis we can be checked by lamp. When we will the cut the cell area of the one side of the egg, we will see the cell sac, and if we small, you can see this in the C portion. You can see the sac. Sac will be open, and you can see the blood vessels, different blood vessels. And in this blood vessel, we can inject the whatever the test atom, and we can see the is this test atom uh, there is a leakage of blood vessels or not, or irritant to the blood vessels or not, or you can say. In the repeat dose toxic study, we are using number of animals. Instead of this using the number of animals, we can use as a computer-based the software to check the effect of the particular drug on the repeat exposure. Microorganism based study. There are two studies mainly commonly done. a ciliate protozoan being used to study the effect of anesthetics on metabolism. A protozoa used as the effect of anesthetics on metabolism, and also the genotoxicity of EMS assay. This genotoxicity or EMS assay is very important. We are doing regularly this assay, EMS assay, when the compound, lead compound in the developing stage, we have to do the EMS test. Is this compound leads to mutation of the mutation in the species, we have to check at the screening level. In this, in this, there are different Salmonella typhimurium strains are used: TA97A, TA98, TA100, and 102. Two amino atrazine used as a positive control for the tester strains with S9 mix. In this, histidine protein is very important. So, one of our experiment, I am showing the results. These are tester strain TA97A, TA98A. This is positive control, and this is a negative control. If you see here are increase in colonies, small minute colony you can see this. That means there is a mutation in the salmonella colony. If you see the test atom, effect of test atom on this colony, there is nothing we can see this as compared to the positive control. So we can do the EMS or genotoxicity assay. The toxic potential of a substance in chemical testing. The toxic potential of a substance can sometimes be detected using relatively simple chemistry-based methods such as HPLC, direct peptide assay. Test worker by mimicking the key step in the development of allergy and binding the proteins found in the skin of the substance. Also, if the protein binds the substance, then it is with index that we call the allergy. Okay? There are different computer-based models are also there for the testing of the as an alternative for the animal studies. Computer-aided molecular drug design, quantitative structure-activity relationship, computer-assisted learning, computer or mathematical analysis, microfluidic chips, DNA chips, organ-on-chips, human-on-chips. We will go one by one. This is one of my PhD work. This work I had carried in Madras Technical College, and we had done some molecule we had tested, curcumin and vedolactin. It's active ingredient of the ethyl talva and curcumin. But in this study, without use of animals, before doing the animal study, we have to check the is this will work on our whatever the disease model. We have to check. Then first we have to do the in silico interaction. What is in silico interaction? So, whatever the active ingredient, we should know the chemical structure and formula of that compound. And this is the curcumin molecular formula, and this is the molecular formula. And then whatever the disease model, I have carried the research in the liver cancer. So in that, which is the protein is high, we have to check, or which molecule is responsible for the proliferation of liver cancer. We have to check. We have to decide, and we should know the structure of their molecules. I have worked on this alpha fetoprotein and metadherin protein, MTDS protein. So we know the test atom molecule, and we know the protein increase in the disease model. So if there is a drug discovery in software is there, we should interact. We should interact, and then we will get these results as per Lipinski's rule. Lipinski's rule. So, what is the PMF score? Is the DOC score, internal binding energy, hydrogen bond length, and amino acid index. In this, we can say that if there is, if they follows Lipinski's five rules, then we can say that this is a good lead molecules. This is the interaction of the alpha fetoprotein with beta-lactam, beta-lactam, or with the protein. This is the result of the interactions. This is also interaction. Computer assisted learning. There are two software developed by Jipmer India, Xcology, XFarm and Xcology. XFarm contains program effect of drug on the habitat. We can check in this by software. Bioassay of history using benefit billion. Effect of drug on the heart. Effect of drug on the blood pressure and heart rate. Effect of drug on ciliary movement of the frog. Esophagus. The user can conduct the experiment and collect the data. Each program can be run in two models, the tutor mode and examination mode. Another is a digital frog. In this, we can look into the different organs and also the incision techniques for surgery and all. Computer program programming we can be also done by the computer programming simulates the hemorrhages, fractures, amputation, and burns also. In in military training, also used as a trauma model, combat trauma patient simulator similar to the trauma. QSAR technique, qualitative structure assessment relationship for the drug development. In QSAR is a statistical approach that attempts to relate the physical and chemical properties of molecules to the molecular activity. Various discussion like molecular weight, number of rotable bonds, log P, etc. are commonly used. Mainly in QSAR, approaches are in practice based on data dimension. It ranges from 1D to the 6D QSAR. Non-invasive techniques such as MRI and CT scan also can be used without use of the animals. In this, we can do the blood vessel study, heart tissue study, surface scan of the inside organ, contrast agents, and diagnostic radiopharmaceuticals. Also, we can do by medical imaging. Computer or mathematical analysis. In this, translation of biological effect into the mathematical equation we can put virtual human organs and virtual metabolism program can now predict the drug effects in human more accurately than animal can. Computers design the molecular structure of the drug to target specific receptor. Prostate inhibitors were designed by the computer and tested in a tissue culture and computer model by passing the animal test. So, this is a very important the uh, concept for the alternative model. So first we should identify the disease. Which area are you interested? If you are having some inflammatory bowel disease or model, in that we should uh, search the articles or publications or what is the problem, what are the blood effect in the blood effect in the bloods, brain, tissue, proteins. We should understand the thing and which protein is increasing, decreasing, which cytokine is secreting, which interleukin is secreting. We should check. Then we should isolate the protein and understand which is the protein is more common or which is the gene is responsible at these levels. That's it, by genomics, proteomics, and metabolic pathway. If you know the protein, then you should uh, understand the which are the molecules we can test on these proteins. Then molecular docking we can do, in silico interaction, in vitro study we can do. We can isolate the some cells from the intestine inflammatory bowel disease in this mainly this intestinal mainly colon artery is affected, so we can from the primary cell line we can isolate it from the colons, and we can in silico we can study the effect of the drug, and also by the computer based screening. Nowadays, microfluidic chips. Microfluidic chips means in this they develop with the same physiological human or animal models. There are different chambers and channels are there. We can keep these channels or fill the channels or then and pour the uh, we can pour the different secretion of the body inside the channels, and we can see the effect of the on the effect of the particular cells. So there are there are different kinds of in vitro models we have studied, such as large test, monocyte activation test, keratogenicity by embryonic stem cell test, skin sensitizer, local lymph node assay, skin irritation for CTEX, epidermis, skin ethic, eye irritation test by nuclear reactive assay, carcinogenicity, cell transformation assay, stem cells model. Even in animals molecules, we have done a lot of things: C. elegans, butterfly, honeybee, for ecotoxicity, cell in vitro. In chemical, HPLC, direct peptide assay, in silico, there are two softwares: Expology, Chromoman, computer-aided molecular drug design, microfluidic chips, quantitative structure, and human studies. If you if your drug is good in alternative models, we have to do some animal study. We have to do some animal study. So different tests for this animal study, we have to do the toxicity study. There are different guidelines are there as per OECD guideline. OECD uh, Organization for Economic Cooperation and Development. In this, we have to do the regulatory toxicity study. There are different guidelines online is available. Just leave. Hello. हाँ ठीक है वेट करो मुझे hello. So there are guideline number test guideline number 428 for skin absorption. Test guideline number 428, 430 for the skin corrosion, 431 for skin corrosion, 432 uh, for 333 NRV photography test, 437 for bovine corneal opacity testing, 438 for isolated chicken eye test, and 439 in vitro skin irritation test. In our laboratory, we are doing the 428, 430, and 431 skin corrosion test. So why all these things required to save the precious life of the animals? So there is a as per 3R replacement, reduction, and refinement, we can reduce the number of animals. If we are taking the too much less animal is also a partial loss of the animals for execution of the successful execution of the study, and if too less use of animals is studied is a total loss of the animals. So we should have the minimum number which will give the statistically significant result. So too much should not be used, and too less should not be used. We should have the optimum number to get the result. So, therefore, the OECD had developed some guidelines, and that all guidelines freely available online. We can you can just now we can see. So, in our DCGI, Drug Control Government of India, they have developed the Schedule Y. Schedule Y having lot of animals in each and every toxicity study we have to use like 10 to 15 per sex per group we have to use. But in this OECD guidelines, in this we can check in the complete study we can do. But in as per schedule Y our new currently known as New Drugs and Clinical Trials Amendment 2019. In this, we have to use 15 number four groups. It will take near about 50 animals we have to use. But in this OECD, within 10 to 12 animals, we can complete the study. There are different guidelines are there for acute toxicity, acute oral, acute thermal. Not only this, there are a lot of guidelines are there. Acute toxic means in the acute toxicity there are three guidelines: 420, 423, 425. In this 420 fixed dose method, acute toxicity class method, and up and down method, and skin sensitization, lymph node assay, 429 and 436. Not only this, there are near about 50 guidelines are there. You can check. You can go to the POUG website. Refinement. What is refinement? It then reduces the total number as well as the quality of the data to avoid suffering by good husbandry by understanding what animal needs. Just give me one second. Hello, Aman, security के लिए हाँ उनको थोड़ा आने दो मैं नीचे आ रहा हूँ ठीक है? हाँ बिल्कुल बिल्कुल so there are some disadvantage of in vitro models. There are some advantages also and some disadvantages also. It will not represent the whole body outcome because in the body, there, if we want to study the effect of the liver, there the effect of the different blood vessels going up and down, food and all these things, we cannot be maintained in the in vitro systems. So some disadvantages are also there. Sub-selection, even primary cell represents sub-selection of the natural population. De-differentiate cells taken out of their natural environment with various feedback, signal, and metabolic demand tend to lose differentiation. Long-term exposure maintenance, so far routine solution for long-term exposure. Cell density also, which will require more cell density as compared to the natural solution. And also, it's required special instruments such as oxygen and all. So, why there is need of the alternative models? Because of the use of lot of animals in the science, some laws are there, and also the ethics. So we need to do things. They are intermingled to each other. So we need strictness of the uh, laws, rules, and acts. We have we have to go for the alternative to save the animals and the humans' precious life. So first is law 1960s prevention of cruelty in the animals. In this, section 15 denotes the committee for the purpose of control and supervision of experimental experiments in animals. The central government the animal husbandry department will develop in some CPCC committee. Each and every government issue, they will ask for the nomination for the CPCC. For the institute level, there is IAEC committee. For the use of the animals, we have to submit our protocol to the institutional animal ethical committee. This committee will be in your college. So there is a responsibility of the high management to develop the committee. This committee will be for the due care of animals uh, as per humanity. That experiments on animals are avoided wherever is possible, and proper skills should be there for those who are doing these studies and properly look after the both experiments and suitable records are maintained. In India, there are some also laws are there. In 2001, Indian Council of Medical Research they have developed one guideline to use the laboratory animals in the medical colleges. In 2009, MCI amendment recommends to use alternatives to replace the animal experiment. In 2012, Ministry of Health and Family Welfare bans use of animals in educational institutes. We cannot use the animals for just for the skills, development of skill. And also 2013, UGC guideline for the discontinuation of the dissection of the animals for experimentation in zoology or life sciences is the matter. In conclusion, the research organizations have to plan as per requirement. The study protocol should be developed. Whenever possible, we should use the alternative models and animal testing is integral and safeguard for the human health to abolish the animal experiments, animal rights activities related to the scientific. Thank you. Thank you. Thank you, guys, for patience listening.
Thank you, sir. Thank you so much for the very informative session.
Thank you. Thank you. It's my pleasure to be a part of your new webinar. You now session is open. If you are having doubts, questions, you can ask.
Sir, just a minute, sir. Sir, there are some queries from the participants, sir.
Okay.
Uh, so the first question is it necessary to take permission from IAEC or CPCSCA to do experimentation on zebrafish?
See, there is no need of taking the permission for the zebrafish model. I will tell some other animal. Uh, only the poultry. If you want to do the work on the poultry, chickens, and all, no need to IST permission. On zebrafish model also no need to take the permission. Even invertebrate, C. elegans also there is no need to take the permission for this.
Oh, okay, sir. Thank you, sir. And the next
For
question is, sir,
I will extend this answer also again. If you want to do the study on the rodents, we have to take the permission from the IPC committee. If you want to do the higher animals such as dogs and monkeys, you have to take the permission from CPCC. CPCC is seated in the uh, Delhi headquarters. So for the up to laboratory animals, rodents should be to the IAC and higher animals we have to go to the CPCNCA and for the lower invertebrates and all and also for the fish no need to take the permission.
thank you, sir. And another general question, sir, how to enroll for PG programs or PhD programs in CSIR?
There are entry the PhD enroll the PhD programs. First is through the CSIR net. Lot of basic science uh, a lot of basic science people will take the CSIR net every year. twice the CSI will collect the NET examinations. I think in January and September. This time due to COVID, it's postponed. It's I think September, October it's there. Also, one is through CSI net, second is through UGC net, and third is through ICMR fellowship. All these three routes, you can enroll the PhD in CSI.
Okay, sir. Another question, sir.
Okay.
Sir, can you see on the screen, sir, are there any dummy models of rats and mice available?
Yeah, yes. There are in even when the Pela training, we are developed the this one block models and even a plastic model. Yes, it's available for the surgery, and we had used in our presentation for this there for rat mice surgery stitches. How to take these two stitches of the dummy models, and even in the frog models are also there. Yeah, it's there.
Thank you, sir. Thank you so much. And the feedback for this session is very good, sir. All are saying the session is very informative. And some little bit problem is there, sir, with the voice, but later on it is clear, sir.
Yes. There is a initial there was a problem actually.
Yes, sir.
Thank you.
Sir, you can see the feedback on the screen, sir. We are displaying here.
Yeah. All thank you. It's my pleasure.
It's our pleasure, sir, that you are accepted our invitation. Thank you so much, sir. Okay, okay, sir. There is small announcement for the participants. The feedback link will be available in the decryption tool of YouTube tomorrow, and the link will be active till 6 p.m. tomorrow. So all the participants who have filled their feedback will get the e-certificate for this e-conference.
Thank you for giving your
Thank you so much, sir. Thank you so much, sir.
Bye.
Thank you,
Bye,
sir.
sir. Thank you, sir. Here I am ending up the meeting, dear participants. Thank you so much.